Bioluminescence in V. fischeri by Ginny Aswad. In nature, bacteria called Vibrio fischeri and a Hawaiian bobtail squid, whose scientific name is Eupremna sculpes, partake in mutualistic symbiotic relationships. This means that both of the organisms benefit from working together. The squid provides a habitat as well as nutrients for the bacteria, while the bacteria camouflages the squid by producing light. As the squid hunts in a moonlit night, the bacteria will give off light similar to that of the sky, allowing for the squid to hide from predators. The bacteria, however, do not have the ability to sense darkness. This leads us to one critical question. How do bacteria know when to become bioluminescent? Let's first take a look at quorum sensing. Bacteria use quorum sensing as a form of intercellular communication to coordinate group behaviors in response to cell density. The bacteria's gene expression changes depending on the cell population density, only glowing under certain conditions, in this case, about 10% maximum density. When the cultures have a low population density, no light is emitted. However, once the population density reaches a certain threshold, or quorum, all of the cells start to emit light due to the luminescent gene being turned on. The proteins that moderate this change in gene expression are encoded by the Lux operon. Now that we know more about density's effect on this particular bacteria, let's take an in-depth look at the Lux operon and its role in bioluminescence. The Lux operon consists of Lux G, Lux E, Lux B, Lux A, Lux D, Lux C, Lux I, and Lux R. The Lux I gene codes for a quorum-sensing molecule called an autoinducer, and in this case, it is a chemical called acyl-homoserine lactone. Once the autoinducer is produced, it will quickly diffuse into the surrounding environment, not having the chance to further interact with the Lux system within the same cell. While this is occurring, Lux R is producing a regulatory protein. This is where population density plays a critical role. If the density is low, the autoinducer concentration is too low to have a major effect on the bacteria. If the density is high, however, there is a greater chance of having an increased amount of autoinducers outside the cells. This increases the likelihood that some of the autoinducers will re-enter the cells and bind to the regulatory protein LuxR. Once this occurs, the LuxR autoinducer complex will bind to the Lux operon, activate transcription, and further enhance the production of the autoinducers. This process is an example of a positive feedback loop. Luciferase is the enzyme that is responsible for the production of light. The products made from the Lux A and Lux B genes make up the enzyme. The luciferase substrates contain flavin mononucleotides, FMNH2, oxygen, O2, and a long-chain fatty aldehyde represented by RCOH. When FMNH2 is oxidated, the excess energy is released as light. This is the light that will go on to help camouflage the squid at night. However, since this process requires energy, the bacteria will only express this particular gene when needed. Now, let's try to answer the question that was presented at the beginning of the video. How do bacteria know when to become bioluminescent? Okay, so over the past few minutes, we discovered that Vibrio fischeri use quorum sensing to detect the cell population density and ultimately the concentration of the autoinducer acyl-homoserine lactone. When the concentration is high enough, the autoinducers will further enhance the expression of the Lux operon genes, resulting in the production of luciferase. Luciferase then catalyzes a redox reaction, resulting in the light that protects the squids. This is how the bacteria know when to become bioluminescent. Now that you have learned about another example of glowing organisms, you have the ability to make an informed hypothesis concerning the mystery of Rudolph's glowing red nose.